This week is all about sensory physiology. In this video, we will be talking about sensory receptors and cutaneous sensations. In the next video, we will talk about hearing and vision. So let's start out with sensory receptors. Sensory receptors are able to change, aka transduce. Transduction means to change. They're able to change environmental energy, environmental info into action potentials, because action potentials is the energy, what's the word? It's like the energy currency of the body. So action potentials are the energy currency of the body. You're changing what environmental information we have and changing it into something that our body can make sense of. So these different sensory receptors are going to respond to a different type. This is called a modality. So it could be pressure, it could be light, it could be vibrations, it could be chemicals, and that would be the modality of these sensory receptors. The modalities are perceived as different because they are going to stimulate different pathways within the central nervous system. They're going to innervate different parts of the brain and be integrated or processed in a different way. Sensory receptors can be just the simple dendritic ends of a neuron, or they can be more specialized. They can be specialized endings of neurons or even specialized cells. So here are some examples that you can find in skin. We'll talk about these in just a second. There are different types of sensory receptors, and they're named based off of the type of stimulus that they transduce. Chemoreceptors are going to respond to chemical stimuli. Photoreceptors are going to respond to light energy. Thermoreceptors are going to respond to temperature. Mechanoreceptors are going to respond to mechanics. So that could be like pressure. And the way that our cell identifies this is by the change of the cell membrane, so the deformation of the cell membrane. Nociceptors, noci has to do with pain. And so these sensory receptors are able to identify tissue damage because there's a signal when tissue damage occurs, and then it will cause pain. Proprioception has to do with like the position of the body, and so proprioceptors sense the position of the body. They can also be categorized according to the location. So a couple examples of this: cutaneous, like your cutaneous membrane. Cutaneous receptors are near an epithelial surface. So this could respond to touch, pressure, temperature, or pain, or stretch even. Special sense receptors are part of our special senses. So hearing, sight, equilibrium, taste, those would all be special sense receptors. There are two types of sensory receptor responses. The top one here that we see is called a tonic receptor. And as you can see from this picture, as soon as a stimulus is applied, there are action potentials occurring, and they're occurring at a regular rate until the stimulus is withdrawn. They do not adapt to the stimulus whatsoever. If the stimulus is applied, you're going to have action potentials firing. If the stimulus is withdrawn, it is going to stop the action potentials. The second type here are physic receptors, and these are considered adapting. They adapt quickly. And so say, for example, a stimulus is applied here, these action potentials are occurring, and then notice the stimulus is still applied, but what happens is the action potentials actually start to slow down. And then when the stimulus is withdrawn, it will stop. This is adaptation to the stimulus that is being presented to the body. So an example of this is smell. Like if you walk into, somebody's room and it smells bad, or Yeah, it has a strong smell. You might notice that right away, but then your sensory receptors are going to adapt to it. The action potential is actually slow firing. And so, if you've left your house for a while and then come back from a trip or something, and then you're like, wow, it smells different in here. I've never noticed the smell of this place before. It's because of this physic receptor. The law of specific nerve energies is just talking about how each type of receptor is going to have a normal stimulus that is going to activate it. This is also called the adequate stimulus, and that's because it is the least amount of energy to activate its receptors. So let me give you an example. Photoreceptors. The normal stimulus or the adequate stimulus here is light. So when you have light, when there is light stimulus, photoreceptors are going to be able to take that information and change it into action potentials for your brain and nervous system to process. But what if somebody were to get punched in the eye? If you've ever gotten punched in the eye before, you may know that intense pressure on the surface of the eye can also cause light. So that intense pressure is also stimulating the photoreceptors. And if you've ever gotten punched before, you feel that intense pressure on the sense of the skin, but then you may see a burst of light because those photoreceptors do get activated, action potentials do get sent, but this is not the normal stimulus, and this is requiring more energy, right? There's more energy within that pressure than there is in maybe a small amount of light within to activate these photoreceptors. So here in our sensory receptors, we don't call them EPSPs. We call them generator potentials. So they are exactly the same as EPSPs, but in sensory receptors, they're called generator potentials. And so if you can see from this graph, they're going to occur at the dendrites again, and they are going to depolarize the membrane. And if there is enough of a jump so that it reaches threshold, then an action potential will occur down the segment of the axon. Generator potentials within sensory receptors are proportional to stimulus intensity. So the more intense a stimulus, the more depolarizing a generator potential is, similar to our EPSPs, right? However, after threshold is reached, an action potential is going to fire, and now the action potential frequency, not intensity, is proportionate to the amplitude of the generator potential. So in physic receptors, the generator potential adapts to a constant stimulus and quickly diminishes in amplitude. So again, these are the fast adapting types here. They are going to quickly adapt and diminish if there is a constant stimulus. So smell is an example. Touch is another example. So think about maybe your butt sitting on your seat, right? That is a good example of your physic receptors within your skin of your bottom adapting to the stimulus of the pressure on the seat. Tonic receptors are slow adapting or not adapting. And so these generator potentials do not adapt. And so here we have a stimulus. It's going to create a small depolarization. Here's a larger stimulus. It's going to create a bigger depolarization. And then we have a greater one. Oh, and there are action potentials firing because we've hit threshold. And now we have a greater one, action potentials firing. And notice as soon as the stimulus stops, the action potentials stop as well. A good example of tonic receptors is pain We do not adapt to pain well. Okay, on to cutaneous sensations. So cutaneous sensations are going to be near the epithelium. So they include touch, pressure, heat, cold, pain, stretch. One interesting thing about cutaneous sensations is that cold receptors are actually located more superficially than warm ones. Pain is mediated by those nociceptors, we introduced that word, and the neurotransmitters used in nociceptors are usually glutamate and this substance called substance P. It's named substance P after pain, so substance P is the pain neurotransmitter. Maybe you've heard of this chemical before. It's called capsaicin. The capsaicin is the spicy chemical in chili peppers. And so hot or heat elicits pain through these capsaicin receptors. So most free nerve endings mediate hot and cold. Merkel's discs and Ruffini endings are going to mediate touch. They are free nerve endings and they are slow adapting tonic receptors. So they record pressure or vibration, or the sustained presence of pressure on the skin. So these are not adapting. These would be different from those physic receptors that we spoke about earlier. Those physic receptors would be these ones. These are called Meissner's and Pacinian corpuscles. I circled them right here. They adapt quickly, so they are physic, they are encapsulated, and they also mediate touch and pressure. But these are going to adapt to continuous stimuli, so like your butt on your seat. So each sensory neuron has a receptive field, which means they have a field of skin that they are going to innervate. So if there is stimulation on these parts of the skin, it is going to change the firing rate of that sensory neuron. These areas are different depending on the different regions of the back, and the area varies inversely with the density of receptors. What that means is if there are less receptors, the receptive field is going to be bigger. So example of this would be the back. The back of your body, like the back of your trunk, have a low density of sensory receptors. That means there's not as many compared to other places, which we'll discuss. And so what that means is the surface area of your back is pretty large, right? And so the receptive fields of each sensory neuron should be pretty large too. Let's compare that to your fingers. Your fingers have very high density of receptors and the receptive fields are very small. There are ways that you can test this out, and I think you will be doing this in lab. A good test of this is to take, you can take something like this, a compass, or you can take, a pair of chopsticks, and you can do the 2-point touch test. There is a 2-point touch threshold, and what that is the smallest distance that 2 points of touch can be perceived as separate. And so on our back, for example, right here, that has a lower density of sensory receptors. Say there is one sensory neuron shown here. This 2-point touch is all within the same receptive field. And so what you are going to perceive is only 1 point of touch. Compare that to a higher density area with higher density of sensory receptors. This is just showing two with two separate receptive fields that are going to be innervating two separate receptive fields. And what you would perceive are 2 points of touch. So this table shows you the different thresholds for different regions. And you can see that on the back, on the thigh, on the calf, the 2 point touch threshold is very high. What that means is 48 millimeters isn't even a lot. It's what, 4 centimeters, 4.8 centimeters. And what that means is that your calf will only be able to distinguish 2 points if it's at least 4.8 centimeters away from each other. If it's any closer than that, it'll perceive that it's just 1 point. And then look at the fingers, they're really close together. And that's because there's a higher density of sensory receptors there. Oral inhibition is when you have a blunt object to the skin and those sensory neurons within the middle are going to inhibit the more lateral neurons. So even though there is a lot of stimulation from both sides over here, there is going to be inhibition over here so that what you feel, what you perceive is mostly within the centered area.